pleasure to. I think there's something on with the sound there. That should be one of the laptops. Can you just mute this Mac? The Mac?
Oh, it's my Mac.
Now let's see. Yeah. No. Yeah. Now. Yeah.
No, I don't know how. I don't know how. I don't know how he set it up.
Yeah, he should be there. Or I can, if I let me see. Let's see her here.
So my
yeah, no, you mute that.
It speaks here.
It's here.
Yeah, I don't know. I have my volume control completely down. I don't have any microphone. I don't have any video.
In the Zoom, can you mute the Zoom? Perhaps
it's muted now.
Okay. We have echo, so apparently the Mac is still okay.
So now finally it does. It works, no?
Yeah, it's because I don't know why, but in some Apple devices, when you rip out the speakers, they are still sending some signals. So
okay. So now hopefully.
yeah, but if you want, I can this one first.
But then if you remove that, then nothing else goes wrong.
Just 1 minute.
Let me, I mean, we can use the time just to make the introduction, if you agree, Steen, so that we don't delay too much.
Sorry about that.
So I was saying that this is a real pleasure just to host Steen here. So I was going to say that Steen Rasmussen, he's a physicist, but I think that's too limited, right, for the activity that he has been developing during all his research career. So let me say that he's a physicist, at least partially by training, right? So he's currently the director of the Center for Fundamental Living Technology, if I say that correctly, in the University of Southern Denmark in Odense. He's also an external research professor in Santa Fe Institute in Santa Fe in New Mexico, where actually he spent quite a long time, but he also spent a large part of his research career in Los Alamos, where he was one of the founders of the scientific field of artificial life quite a few time ago. So I think that's probably one of the main activities of Steen Rasmussen during his career. Although he has been also contributing to many other different fields. And well, I was surprised to see that he also contributed to things like transportation, for instance, or disaster mitigation activities to optimize this kind of, let's say, activities, applying some of the physicist training to that. So Steen will be here, I hope that quite often in the future. So he will be paying us some visits. And we are also really happy and proud to say that he will be also contributing to one of the outreach activities that we have at the IPC. So he will be contributing to the program of art and science that we have created in collaboration with Tabacalera. So we appreciate very much your visit here, Steen. We are really thankful for your contributions to the IPC and to this particular project of art and science. And thank you very much also for this talk that you are going to deliver today. So thank you, Steen.
Well, thank you. Thank you for inviting me. San Sebastian is a spectacular place. It's a very, you already know that, but my wife introduced me to this place here a year ago, and then, sort of got the ball rolling. So my main activity and my main love, scientific love, is trying to understand the creative forces in nature. I mean, as a physicist, why are we here? How can life emerge, and how can life develop, and how is all this complexity possible? There must be some physical principles that hopefully we can understand. And I've done this for many years, and I want to make sure that I acknowledge some of the many wonderful collaborators that has helped me along the way. I wouldn't have gone anywhere without this big team of people that I both have had in the US. I spent 20 years at Los Alamos National Lab, and now I've spent 12 years up in Denmark, and we've been fortunate to have great teams and also to have a good funding every once in a while anyways. So my plan is to, now we're not that many, so, okay. Oops, yeah, thank you. Yeah, this is better. So I think we're all looking forward to that. I mean, in Denmark, they removed the masks a month ago, and everybody was smiling. So I'll try to address some of these things. First of all, when we talk about life and creating life, then we have to figure out agree on what life is. I will give you my perspective on how I think the most likely emergence of life happened on Earth. And then, of course, the most important thing is how can we, as humans, recreate life in the laboratory? We haven't done it yet, else you probably have heard more about it. So there are some open questions, and I will discuss some of these open questions, and I'll solicit help from you guys to maybe figure out how we can get to the end and actually make artificial life. Then, if we want to make artificial life or artificial living materials useful, we need to evolve useful properties. And this is a non-trivial thing, which I'll spend a little bit of time on talking here at almost at the end. But of course, the big vision is, at least my vision, I think there's some really incredibly interesting things that we can do as humans and to preserve the planet if we can really make technology that's based on living materials. So artificial life is the study of human-made life. So it doesn't explain what life really is. So as far as I'm concerned, there's no difference between life, the sort of natural life, and the life that we can create. So my operational definition is that life is a physical process that uses free energy to transform resources into building blocks that makes a system grow, divide, and evolve. The growth and division is in part controlled by inheritable information that can change, so selection and evolution is possible. So in my view, there is a gray zone between life and non-life. It's not a sharp definition, and this is somewhat controversial. There isn't a complete agreement in the scientific community, but nevertheless, this is what I'm going to use. And I think that, or not, I think what this study is trying to address is whether there are limits on materials and systems that can support living processes. I think this is a very important scientific question. That means that it's not only biologists that really should be addressing this. And in this session, we're going to talk about wet carbon chemistry. Could be robotics, it could be living process in cyberspace, or it could be some ecosystems that are a combination of these three here. But and even though I've made a lot of work in these other areas, we will only talk about life that is based on wet carbon chemistry. I know, I put this in parentheses because I do believe that we can have life based on other kinds of chemistry, and we have Mark down here, who's making lifelike systems with gold spheres, and I think you can do it with many other systems too. So, in my opinion, the most likely origin starts with a free energy gradient and a catalysis. And I know you all know what catalysis is, but catalysis is really a modification of the rate at which some reactants are being made into products. So if you have catalysis, you can have a selection, or you can have a preference for certain products over other products. And that's actually the essential feature of all metabolisms in living systems. You have again your what's it called, your free energy gradient, and then you create you have some free energy, and then you can take resources and make building blocks if you have catalyst. And it's important also to include this environment, because the environment becomes very important later on. So this is sort of the core of life, and this happens in nature and geochemical systems also today. Now, one of the key steps is that these building blocks that are being made, they should actually act as catalysts. And then we have autocatalysis. That means you have a positive feedback. So the more of the catalyst you make, the more of the building blocks you can make. And part of these building blocks, they make up the catalyst, and therefore you have something that is self-reinforcing. Now, here is another very important sort of jump. I believe that at some point, this catalyst could have different states. It could have different shapes or different functionalities. So as you made these building blocks, they could combine a little bit in different ways, so that there would be it would be possible to have a selection of certain catalysts. And then, of course, the difficult question is, if it's catalyst number 2, that's the best, will it then also preferential make building blocks that can make catalyst 2? And these are some of the questions that we will try to address. Now, maybe, yeah, let me just jump to it. I mean, at this point in time, we don't really have a discussion of information. I know many people, they associate life with information, and I completely agree. Information is really important, but that's a secondary thing that comes in exactly at this point here. And if we look at a system that has W states, then we can use Boltzmann's fundamental equation to see how many different combinations we can have, and this will then tell us the entropy, or how much information that's possible to encode in this catalyst, if it can be in W different states. And we have to be careful here, because usually when we talk about information in connection with life, we think about a sequence, an RNA sequence, a sequence of peptides in a protein, or DNA or something like that. And this is called sequential information. But when I talk about information, I talk about compositional information, which is more fundamental. And this is Lancet, at least as far as I know, and his crew from the Weizmann's Institute that first proposed this definition. And the compositional information is then a superset. So the sequential information is a special case of this kind of information. And I believe that we really should be focusing on the compositional information, and at least to begin with, forget sequential information. Now, many of you, or some of you, the physicists in here, are probably familiar with Landauers' principle. And it says that if you want to create information, or modify information, or delete information, you need free energy. And you can actually quite easily calculate using Boltzmann equation and the fundamental relationship between entropy, energy, and the absolute temperature. Then with a little twiddling, you can figure out how much energy it takes to create 2 bits. I mean, a 0 and a 1. And if you then put in Boltzmann's constant and the temperature of sort of 20 degrees or something, then you get a very small energy. It's a little, it's about half of a kT for those of you who are working with these numbers, about a little less than 2% of an electron volt. So this is sort of this is a physical proof or indication that you cannot have information first. You must have free energy before you can have information. So therefore, we have to start with the metabolism before we can even start talking about information. So the next step, or if you, at least this is something that also needs to be available. You need to have a. So now you have a. You have this ability to encode your own creation, and this metabolic process together with this catalyst have to be kept together. And we talk about it here in abstracto, a container that have these two processes in close proximity, because you can't have a metabolism over here and the catalyst, the information catalyst over here. They have to be kept together. So therefore, you need to have some of the building blocks. They should be able to self-assemble and make a container, an aggregate of some kind. And then you actually, you're ready. So if you have so what you need to do if you want to make it again in the laboratory, you need to have a metabolism, and you need to have an informational system, a catalyst, and you need to have a container, and it has to be in a benign environment, in a really nice environment that enables you to maintain this system. Because, I mean, as you will see later on, some of the molecules we're using, if we have oxygen, everything stops immediately. So you have to protect these systems. So the environment is is much closer than we usually think about when we think about living organisms. We feel very confident that we are living, and this out here is not living. But I guess if we jump into space, we'll find out that that's not true. The other thing is that to note is that that life, it comes out of all these interactions. Life is not a single, it's not a thing, it's not a molecule, it's not an aggregate. It is something that's generated out of the interactions between these different components. So it emerges out of the interactions. So this is our functional or organizational design that we've worked on for, oh God, yeah, when was it? I mean, it's almost 20 years now. Where we start with the metabolism. We use light because the chemists, they are scared of having too many components in the system at the same time. We have some resources that come in. The metabolism can then make building blocks, but these building blocks, or the production of the building blocks, is catalyzed by the compositional information. And also the information catalyzes the production of building blocks to make the container that can keep the information and the metabolism together. And the container is self-assembling, but then you also need to make sure that the information can self-assemble to the container and the metabolism can self-assemble to the container. And this sort of closed loop organization, I guess Manfred Eichen was the first to really emphasize that. He won the Nobel Prize many years ago in chemistry. In chemistry, ultra-fast chemical reactions. Ilya Prigogine was probably the one that emphasized how important it is to, if you, in context of life, that you have a system that's driven by a flux of materials and energy. And for Neumann, I want to emphasize too, using the inventor of the modern computer that for Neumann design is in all of the cell phones and our computers today. And he emphasized that, look, to have both living and intelligent processes, actually including intelligent processes, they should in principle be independent of the substrate. So you should be able to make life and intelligence in other systems too. So we are standing on top of the giants here. So back in the days, we tried to make some sense of what we were doing and what other people were doing. So we sort of invented some functional diagrams so we could compare the different groups that were in this business. And I think Gandhi was the first one to actually identify metabolism, a container, and an informational system. I think the most famous Protocell proposal is the one of Shostak, Bartel, and Luisi, where they talk about the RNA. The RNA world is, so to say, the fundamental one. This one is where you actually have bits and pieces from modern cells. You have a translation machinery. That's what most money is put on. That's where most people are working. Well, it turns out that, well, this is our design here, which we'll talk much more about. But it turns out you can actually use these diagrams also for robotics. Here we have a small ecology of self-reproducing robots. It's not so easy to make such a system. I mean, nobody has done it yet, but we certainly have designed one, and this is of course of interest, sort of scientific interest, but also if you want to send people to Mars or on the moon so they can live off the land, you need to have a self-reproducing machine that can, or machinery that can take care of you and everything else. And you can also use it for living processes in cyberspace, but we don't have time to talk about that. So this is the essence of our protocell. This is the engine. We use ruthenium because that's the best known chemical or photoactive chemical compound. It has a very long excitation time. So when you shine light on a ruthenium complex, this is ruthenium-2 bipyridine, tribipyridine. You can knock an electron from the ruthenium out to one of these organic compounds. And if you don't do anything, it'll then jump back again and it'll shine light out. But if you can deliver an electron to the ruthenium here, then you are in a situation where you, for instance, can take a precursor. We've used picolinium ester, and this molecule together with this precursor molecule can make fatty acid and then some picolyl, some waste. And we can actually also use it to glue together to ligate DNA strands. So this is some experiments. You can see different configurations of the catalyst. We don't have time to, I think, go into all the details, but fundamental idea is you have resources. In this situation, these are an oily substance that is converted into fatty acid, decanoic acid. And down here we have an oligomer, a DNA oligomer that can be turned into an active oligomer. This is a precursor. You have to deprotect it using exactly the same chemistry, and then you can make longer DNA polymers. So these are the experimental results where we show over time you make more fatty acid, and over time you make more of the long DNA. So it took a long time for me to understand this. So if you are a little bit confused, if you're not a chemist, then don't worry. But I just wanted to show you that that's where this is the center of what we are doing. And of course, we could exchange ruthenium. It's kind of a pain to work with, but now ruthenium is working, and every time you change a molecule, it usually takes years for you to get your experimental system to work again. So we don't dare to shake or rock the boat. So this is our first real experiment that demonstrated that our idea was okay. We have time here, and this is the amount of fatty acid. This is the fatty acid production. And what we see here is that we get, if if there was no influence of the emergence of these aggregates, then we would have gotten, this is a reaction kinetic analysis of, we would have gotten this curve. But because we have an impact, it impacts the kinetics, whether you have aggregates or not, whether it's a homogeneous system or heterogeneous system. And therefore once you have you reach a critical vesicle concentration where these vesicles are made, then you rapidly get a larger surface, and that means the rate of the conversion of oil into surfactant or into soap goes down dramatically, and at some point it sort of levels out. And this one is this is a background reaction. So our resource molecules, they will slowly decay in water. I mean, water, as you know, is a very aggressive solvent. So this is, so to say, this is a death curve. So anything that can survive will have to be above this death curve. We'll talk about that later. This is a very busy slide. I just wanted to show you that this is our reaction diagram. We have the ruthenium engine over here, and here we have our electron donor. This is oxoguanine. It's a form of guanine that has an extra oxygen, therefore it has that name. But unfortunately, when it gives the electron away, it actually loses a hydrogen. And this is not so good because it will then break down. So you need to repair it, and therefore you need to have a proton donor, a hydrogen donor in the system too. And it turns out this is a fast process, so all of these things are working. I don't have time to go through all the details. The reaction kinetics is out there, and if you want, I can give you a reference to where we do it. But I want to emphasize we have three different numbers here. This is the one we're going to talk about, the quenching rate here. How can we control that with compositional information? And this is so to say the death rate. This is the background hydrolysis of our resources, and this is actually the current rate limiting factor. We have a rate that's a little below, it's about 1/2 per second, which is very slow. We can calculate this rate rather precisely because we're using a laser, and we know how much energy, and you can then do the quantum yield. So this is the rate limiting factor here. So we can't get, so if you want to go and change this one here, which is very fast, we need to slow it down a bit. It won't impact the production very much, but if you want to have a selection of this kind of information, this compositional information, we need to slow down the rate. Just want to show you this. We have not been able to reproduce it. This is actually a simultaneous production of fatty acid and longer DNA strands, and we haven't been able to quantify it. We've made quite a few experiments, and it was Kate who was in charge of that. He's very capable postdoc who went off to industry and have a happy life. But this is something we have demonstrated, so we can actually have both processes in one pot. This is also a little bit of a historical picture. This is our first postdoc, Sarah Maurer, who demonstrated a life cycle. This is our first life cycle. Here you see a mixture of fatty acid with the ruthenium complex and with the oxoguanine. Again, it's not on a DNA strand, it's just oxoguanine that has attached a tail. Then you homogenize it, she squeeze it through a filter, and then you feed the system with the oily substances. These aggregates, they are some 100 nanometers. They're below a micron, and here they become several microns. And then you put light on, and then you'll have these big aggregates. They'll break up to smaller ones. It took a while for us to be absolutely sure what was going on. This was Pierre-Alain Monard. He suggested that we could actually put beads inside. I mean, there are many, we've done gazillions of experiments, so I'm just going to mention a few of them because we got, criticized from referees that, well, can you actually be sure that you have budding and you really have generations? And we do. You can see here, we do get budding. We start with beads, and then we end up with just as many of them without beads, and they're coming off like we see here. So far so good. Now the thing that has slowed us down was the ability or the inability of us to replicate the catalyst. And again, now we are using. There's a long story of why we're using DNA. The very short version is that I can't afford to. I wanted to use a peptide nucleic acid because this is a molecule I like a lot, but it's just too expensive. I didn't have the money to have a protein synthesis lab. So, and RNA is too nasty to work with. It's difficult to work with. If you sneeze in the lab, then it gets upset. So DNA is very robust. It's very cheap. And you can buy it you can take it off the internet. So we decided to go with DNA. So the strategy is that, again, this Protocell is a quite an unusual design where we have this is our container. Then we have our DNA molecule sitting here, and we have our ruthenium molecule sitting here, because we want to use the container as a two-dimensional container, because in that way it's easy to get resources in and to get waste away. All this monkey business of having, the good stuff on the inside here, then you need to have ways to penetrate the membrane, and I don't like that. This is much simpler. This comes later, but if you want to have a minimal living system, I think this is the way to go. You can disagree because this is how it is in modern life, but I think there are many things that are different. They were different in the beginning than they are here today. This is simply to make it simpler. So we need to replicate DNA on the surface. And remember, DNA is only used because we can incorporate oxyguanine, and it is the DNA's redox ability that we are interested in. We don't care in principle what the sequence is, unless the sequence influences the redox abilities. And the redox abilities or the redox potential is determined primarily by the oxyguanine, because oxyguanine is a molecule that can deliver an electron. The other things, unfortunately, that's thermodynamically impossible. So we have published a lot on that, and we have made a bunch of experiments. We've shown that this is actually Ebola. We had my center is called Flint, and this was number 12. We had many of them. I think we had almost 20. Number 12 is a nice one, and we anchor the DNA nicely. This is a single, very long one. And here we see this is with Mashiaki Imai from Japan. So we are working together with, he's actually doing, this is not a fatty acid vesicle, this is a phospholipid vesicle where he is demonstrating what's it called, amplification on the surface, but it's not going fast enough. And I think the reason is because, and this is also what we were struggling with for years, and many graduate students were very unhappy. It was because we weren't as smart as Julie Gibbs Davis, because she figured out that if you want to prevent product inhibition, you have to be a little smart about it. Product inhibition is when you have, and it's kind of easy to understand. If you have a small piece of DNA that hybridizes to another piece, it will fairly easily fall off. But you have two very long DNA pieces. They're almost impossible. They're so nicely or so strongly glued to each other that they will not separate. So you can imagine if you put pieces together to make a plus and a minus strand. So once you have made the plus and the minus strand, they will not separate. That means you cannot make new minus and plus strands. So how can we get around that? Well, she said, look, what you need to do is you need to use a so-called lesion-induced DNA amplification method, where you have some mismatches, and then or air bases she used, but we can also have an extra base, so you make a nick on the DNA, and this bulge makes the double-stranded much less stable. And here's the key to understand or to investigate these systems. This is what we've done every time we've made these experiments. First, we try to see what we can do from a computational theoretical perspective, so we don't waste too much time for the chemist. So the connection between thermodynamics and kinetics is given by this fundamental expression, where you have the forward rate and the backward rate. If you divide the two, you get the equilibrium constant, and that's given by the free energy of the reaction divided by, if it is measured in divided by the gas constant times T. And it turns out that pretty much all the forward rates are about 10 to the 7 or 2 times 10 to the 7 per moles per second. Or actually, this is wrong. I think it is the second moles. So I'm sorry, that's wrong here. So we know this one, so we don't know this one. And since we can measure the what's it called, delta G, either we can go in Santa Lucia and Higgs in their famous 2004 paper, they have these things tabulated, or we can use NuPack that will actually give, if you have a sequence, then it'll give you delta G. So we know if we know the sequence, we know delta G. Since these small sequences, they all have about this on rate at a room temperature, we can directly get the what's it called, the off rate. So now we can, I should say one thing here. I really appreciate Joyce's work, and I was very impressed when he's in 2009, where he was able to make a self-replicating system, and we were not. And I spent a lot of time talking to him and studying what the heck he was doing. But I realized that this is actually, he was before Gibbs Davis, but it's exactly the same. When you look at the free energy of this one and this one, delta G for this plus delta G for this is pretty much the same as delta G for these two, because of these, he's not using bulges, but he's using these stem loops. So it's the same way of preventing what's it called, product inhibition. It's the same thermodynamics that he's been able to beat. And they did that with I don't think they, actually, I don't know, I don't remember anymore, but they found several families that could do this here. So what we did was something else. We said, okay, before we do the experiments, let's do simulations where we send the input of the simulations, the parameters, to via what's it called, an interface, a web interface, to one of my colleagues, Norman Packard's AI-based design of experiment algorithm that's over in San Francisco, and on his servers there. It will then make a map from our input parameters to what the resulting rate is. If it has enough input from, we've given them some input parameters for the simulations and also given the results. The result is the performance of the replication. And this is one of my students, Ditlev, that did that in a master's project. And no, actually, this was in a bachelor project, and he later did a master's project. I'm very impressed. He has a background in industry, so it was very easy for him to make this interface. But else, it works by designing the what's it called, the surface here. This is just for 2 parameters, and you want to go to one of these maxima. This is just a pretty graph, usually they don't look like, they're not as complicated as this one. And you can actually use that for your optimization processes. So these are runs from using the leader technique. And this is the first generation. We have a bunch, I think we have about 100 simulations here. And this is the this is the response time. This is how long time the simulation took. If it takes longer than an hour, we cut it off. And as you see, as we go through generations, the system both explores the parameter space, I mean, by varying the different parameters in the simulation, but it already in the second generation, it discovered the fastest combination. So this is, I think, a really beautiful way of doing these kind of things. And so again, this is this is Norman Packard's algorithm. Another nice thing about this algorithm is that you can ask it, so which are the rate limiting constants? And as expected, it is this is the one that is most important. This is the product inhibition rate. Over here, we also have the next one is the ligation rate. We wasn't aware of that. We knew this one, but this one we weren't aware of. And actually, if we twiddle with that a little bit, this one could become more important than this one up here. But we don't know how we can increase the ligation rate using the chemistry that we have. So this is this is the main thing. This is for a ligation rate that is less than 0.1 per second. So here we have the examples. Here we have the measured curve by Gibbs Davis. This is just a test where we try to see whether we can get we can reproduce their experimental results. And the green one is the most convincing. Well, we don't understand why we can't get it to follow it more closely. But there is a difference between reality and mathematics, as we all know. So this is the fastest one we get if we keep the parameters, the physical parameters that we believe we have. That's possible. And this is if we allow what's it called, ligation rate that is a little bit faster. So then we can get a replication that is in 5 minutes. That's very, very fast. But I think that's unrealistic. But it still is interesting to explore. From a theoretical perspective. So now we already know from our studies that depending on how the compositional information, again, this is it's the free energy, that it's delta G that really determines how fast this catalysis runs. That's a dominating factor. The other thing we need to figure out is how fast will the metabolism run, depending on where you place the oxyguanine. Because in our studies, we actually had the oxyguanine sitting on an anchor out here. We did not put it into the DNA yet. We have tried to do it, it didn't work. And there are all kinds of reasons for why it didn't work. But this is, I think, at least this is a system we believe the most. I have a graduate student, Christopher Thompson. We've discussed that here the last week. And I made this figure last night. And we have, I'll show you some curves that he did last night also, or this morning, I guess. Because what we believe is that if you look at it, so this is something I don't know much about, and I hope that some of you could help me. And it's charge transfer. I wasn't taught anything about charge transfer as a physicist, and Christopher, he's a sad student. He's also just a physicist. And the people that I know, they don't know enough about charge transfer, so they don't dare to say how we should really calculate the charge transfer rate along a strand. But then we figured out that Moser and Dutton, they are great people. They have studied charge transfer in biological systems, and they're using sort of a empirical formula that's based on quantum chemistry, but still it's much easier to use than these fancy other methods we've tried. We've spent several months trying to figure out what to do. But here you can see that if you have a charge transfer in vacuum, you have a very fast. This is a log of the rate, the reaction or the charge transfer rate. It goes fast. It goes down very, very quickly with the distance. And if you have a pi coupling, as you do, if you to some extent you have in DNA, well, it's not zero, but in principle, these are sort of the two boundaries. If it is in solvent, he believes that 0.4 per angstrom is a good number, and it's covalently bound, then it's 0.7 per angstrom. So these are the results here, where you have the distance in angstroms here. We're using Moser-Dotton's approximation here, where this is the decay with the radius. And remember, here's log, so this is all, it has to be, if you take the 10 log or log 10 on, or sorry, the exponential, the 10 exponential up, raise it to the 10th power, then you get, no, I didn't say that right. It's not the exponential function, but the, what's it called, 10 to the x or 10 to the k, then you'll get the right, you get k directly. And this is a free energy, this is the redox energy, and this is a so-called reorganization energy that is involved in organizing the molecules so that you actually can have the process to run. And here we have, it would be our system is about 18 bases long. So if you have a 18 base long, we should start in the other end. If the path is only one base, then it's lightning fast. And I don't think that you can have, this is again the log up here, I don't think you can have this fast reactions even with electrons. So this one is, I think that then we go outside of the what's it called, the range here for this approximation. But you can see as you go, as you make the path longer and longer, where the electron has to pass through the DNA, you become slower and slower. This is a rate constant, the estimated rate constant. And as you make the concentration of the ruthenium lower and lower on the surface here, so the average distance out to the electron becomes longer and longer, you'll also get a lower and lower performance. And it's interesting that so what we one of the parameters dominates here. This is how we have expressed it. We've never tried to do this before, so and it hasn't gone through peer review. So I hope you guys will tell us if we are screwing up or we're doing something wrong. But this is how we believe at this point in time. So the question is there anybody here, or do you know anybody maybe in the internet here that can help us with a charge transfer, to understand charge transfer, in particular, if you want to understand how the sequence, this compositional information of the DNA, how that influences the charge transfer rate. Because why is that interesting? That's interesting because the compositional information for the protocell must both act as a catalyst, and it has to be able to replicate. So we need to have two things satisfied at the same time. If we think we have a subspace of strands, then we need to make sure that the rate constant of this charge transfer is larger than a certain amount. Now I put it to 2 because I wanted to have a little faster than the rate of the quantum yield. And this is the amplification time for the DNA. I said it's a little less than an hour, and that's what was our cutoff rate for our leader system. Then we can, so then the question is, of course, how likely is it to find a sequence from this subspace here that satisfies one condition or both conditions? And can do that with Monte Carlo simulations for both systems here. And then we can simply calculate the probability of one of the conditions being fulfilled by counting the number of strands where the condition is satisfied, divided by the total number of strands we've used. And the same is true for the replication. And of course, for both of them to be true, then you have to count the ones that both things are satisfied. And in my opinion, then comes something really interesting. This was Artemi, one of my colleagues from the Santa Fe Institute, that came up with this proposal here. I've always wanted to make a graph that looks like this here. I have this sense that if you if you have time here, and then you have information up here, then one of the sort of, if you say here we have a self-reproducing something rather, but at some point, the information starts to grow. This point is when, if you want to define something to be alive, is when you start to accumulate information. That's when you have selection that enables a catalyst, the informational combinatorial catalyst, to be better and better. And I think we should be able to do that by simply using the functional information as defined by Hazen and Shostak down here, where you simply take the natural logarithm. This is the standard way to find the what's it called, the information. And then I think we should be able to, at least to begin with in simulation, and hopefully once we believe more or less that we have qualitatively correct answers, then we can go to the experimentalist and hopefully convince them to or pay them to do these experiments for us. So how far are we from making an autonomous protocell? Well, here you see all the black stuff is stuff we have published and we've done, and we've spent about, I think from the beginning, about 16 million euros. And I think we could, with what we know today, we probably need about 6 million more to maybe less if we're lucky, to both have the DNA controlled metabolism to work. So actually to test how you can place the oxyguanine on the sequence, and also make sure that we can get this leader-based replication to happen on the surface. I mean, it sounds, for theorists, it sounds easy, but it's a pain. I mean, so to get these two things to work, then we know that, well, knock on wood, that these two processes, they work together. So then we should actually have our first living self-reproducing molecular machine. Then of course, the next step, this I think is a big deal, because we'll be able to make simple evolution by giving it two different kinds of food sets. And then with what we know about these things, we should be able to show simple selection. Now, if we want to make something useful, then we need to evolve, we need to functionalize this thing. And that's a different story. And we need to take the big guns into use to actually get there. But I think this is to be whoever manages to make the first autonomous living system from scratch. I mean, they can have a big party. So that's why I designed a robot here that I want to use both to make the screening of both screening here and screening here. It's really tedious to do these experiments, and for about $100,000, we can easily make such a system here where you have a robotic arm that runs on a rail, or you can move. So you have a liquid handling robot, and you once you load your system with these well plates, you put it to a detection platform. What's it called? Fluorescence is the easiest, but of course, in the beginning, you'll have to do HPLC, and you have to do other things to make sure that you know what it is you're detecting. You send it to the AI-based predictive design of experiment algorithm. It sends suggestions back, and then you simply run it. You can say, okay, do 10 generations or 100 generations, or until you have a satisfying or a certain performance has been reached. And I think that such a thing could be used for many other things than creating life. I think that many universities would be happy to have that, because it wouldn't cost that much. I mean, if you want to have a self-driving lab today and you go to the instrumentation makers, they charge you 10 millions without smiling, without blinking. Okay, so I don't know, how are we doing on time here? Can I spend 5 minutes on evolution? Because one of the things that in the beginning we of course we thought, well, if we can make a system that can undergo evolution, then it can invent an eye and a bicycle and all kinds of other things. But it turns out that people have tried that, and all these evolutionary systems that we have played with in robotics and in cyberspace, and certainly also the chemical system, they get stuck. You get into an optimization and then nothing happens. So we wanted to figure out what the heck was going on. So we proposed that there might be two different types of evolution. One of them is an optimization process, which I'm going to talk more about, and the other one is evolutionary expansion. And the optimization process you get when the number of components and interactions, the type of interactions you have in the system, is more or less constant over time. For you to have an expansion, you need to add more objects or components or types of interactions. Else you cannot get an expansion of that means you cannot get new properties. So we've studied many different systems. I'm just going to show you a few of them. We start with the biological one because it's sort of more in line with what we're doing. So this is this spectacular experiment that Linsky and his group is doing up at the Big Lakes. Where is it? In Michigan. So he's a cool guy. He's a biologist, and he's had a culture of E. coli that he's been studying for tens of thousands. I think he's up above 100,000 generations. And what he's doing every once in a while is that he gives it something else to eat. So maybe it's peanut butter here, and then it is pate the next day or something. And then he looks at how the average fitness evolves, how it changes over time, and that's related to the size of the, well, anyways, he has a long conversation. There's a long discussion about how you can measure fitness. I don't want to get into that. This is the beginning. Here you have the actual fitness divided by the beginning fitness, so it's normalized. And you see that it somehow levels out. This is in a pure colony of E. coli where you change the food source at this point here. And this is 50,000 generations here. Now, Paulo, one of my colleagues, he said, look, you just have to take the logarithm of the time, and then you get a straight line. I said, are you sure? Yeah. And that's true. Of course, this is not maybe 100% convincing because we don't have gazillions of data, but that seems to be true. And this is a signature of an optimization process. So, and why does that make sense? It makes sense because E. coli can't invent an eye or a wheel or wings or something like that. There's simply limits to how much it can do, and it does the best it can with the things that it has available. It will be completely different if you have a colony, if you have an ecosystem where you could interact with other components and other species, exchange DNA. But once you have conserved interactions, then this is what you get. You won't get anything else. And now some of the physicists might be happy because this is a magnetization. This is a ferromagnet. And here you have magnetization over time. And you see this is also pretty much a straight line. This is over many more, what's it called, orders of magnitude. You can, this is if you look at it as quakes. I mean, this is extremum extrema calculation. Whenever you make a new, whenever the magnet finds a new minimum, and you note every time it does that, and it's a remarkable straight process, and the same, you can get the same results from a if you measure, if you do it in a spin glass. But this is again optimization, and we feel more comfortable thinking about optimization or energy minimization if you have a ferromagnet, right? It's something we know, we learned that in school. But it actually is the same, I believe, as we see in also with technology. We looked at many, we are not the only ones who's looked at that, where we looked at technology. This is photovoltaics, where we again see that once we look at this as a price of per watt of photovoltaics, and it's a nice straight line we get here. This is also optimization. Wrights, not the Wright brothers that originally flew, has figured out that there is this relationship that you have optimization, and it's Wright's law. So this is actually an example of Wright's law. We had to make this exponential or logarithmic, because if you look at the number of photovoltaic units that are being put out, then it grows exponentially. But still, this works for technology too. This is an optimization. Yeah, I think we are running out of time, but there's a few different forms that you can this optimization can take. You can look at our paper. But what I want to say is that we end up with a situation where the fitness or the minimal energy can be expressed as it's actually a function of the number of interactions, and I don't know how you actually calculate the number of interactions in a spin glass or in a E. coli, but you can certainly think about this as a theoretical measure. Now, there's not enough good data in biology, and I'm not aware of physical systems where you have evolutionary expansion. But there are tons of data in economics and with patents data and these kinds of things. So if we take the gross domestic product as a proxy for human fitness over time, we can discuss a lot whether these numbers, actually this GDP per capita, whether how the heck the historians, they are able to measure it. But nevertheless, let's assume that they are. And this is the logarithm. Then it looks like that. This is Italy, Sweden, Turkey, and United Kingdom. You can see how United Kingdom, they were really good with their colonies. And here we have the Pope that was very, very wealthy in Italy. And then everything explodes once you get to the industrial revolution here. So there is a so what you can do here is you can take this, you can actually measure this interaction, the interaction density, or the number of interactions over time, by removing the trend. If you have first taken the logarithm of time, because if you know it is if you know it's an optimization, then it should be a straight line. So everything that diverges from a straight line is then the expansion, and you get something that looks like that, and it means that you actually have something here that is first of all, this is logarithmic here, so it is one of the exponentials simply because of the logarithm here. But it means that we have an exponential growth of our interactions in our in our economy. And this is also what many other people many of my colleagues at the Santa Fe Institute, they're looking at technological evolution, and we get an exponential growth of the number of interactions if you look at it in a sort of an ecosystem or on international scales. So I'm sorry I have to run a little bit fast through this, but let's jump back to the protocell. So what this means is that for us to make life, I believe, we don't need to worry, because living systems, E. coli optimizes when you change food source. So we'll be happy just to have a system that can optimize. So if you want to have a system that can sequester CO2, or that can be used for medical purposes, or sort of your imagination is the limit, we would need to do something else. This is again getting to this here. Let me just get it here. To go out and develop useful systems, we need to introduce new things from the outside, novelty from the outside. That's the only way that the system can learn, is able to recruit things from the outside and learn. So I just want to conclude by my perspective on what the implications would be. I think that, and I don't know, I also worked quite a bit on these cyber systems, like living process in there and intelligent process in there. So I forgot to remove that. But from a scientific perspective, once we are able to create life from scratch, then we will, for the first time since Adam and Eve, be able to address the question why we are here. I mean, what is it that makes it possible for life to emerge? Because we've simply recreated it. So I think that this is a big deal for science, and I think there is a fantastic opportunity for physicists to engage with this, because it is, the question is, what does matter? Why is matter sometimes able to do all these amazing things and make us? From a technological perspective, we have to get rid of this paradigm as soon as possible. The way we use technology, the way we live today, is we take up resources, we build our stuff, and we throw it out. It's very bad. What we have to do is we have to make technology that's based on the same principles as living systems. And I think we need to go down. It's great to recycle, and we should do that too. But once we are down at the material level, where we can actually start to decompose our artifacts, then we will finally be in a place where we can be in harmony with the biosphere. And this, of course, the reason why we've done this is, well, this was long before we had religion, I guess, but certainly we've used the book of the Genesis as an excuse and said we're something special and we're something separate from the rest of the living world, which is, of course, not true. We are part of we are part of the living world, and we have to sort of say adapt to the fundamental laws. And this is a stupid way of going. That we should do that. And I think that these being able to make artificially living materials will help us to make technology that enables us to go there. The last thing I want to say is some kind of an organizational idea. I believe that we should go to CERN and ask them to expand their mission to include the fundamental question of the origins of life. CERN is an expert in origins of the universe, matter, and energy, and they're the best instrument builders in the world. So they could build these evolutionary machines. Of course, it'll have to be a different kind of scientist. The high energy physicists, that they should allow other physicists and other scientists to come on board. And interestingly, I don't know Fabiola, I mean, the director of CERN, but Stu Kaufman, one of my friends and colleagues from the Santa Fe Institute, he had a cocktail. He was at a cocktail party with her. And if any of you have met Stu Kaufman, he's a very charming guy, and he's a great car salesman. And he sort of convinced her that the physics of life is really the future, and CERN should get with the program. And she then asked Markus Nordberg, he's the program director at CERN, to organize a meeting where we looked into how this could be done. We've had a number of meetings. I haven't been part of all the meetings, but there is, so to say, there are gears in motion to actually make this happen. The big Dutch artificial cell project, I know these guys too. There are some of them there talking to the European Commission to make a center for artificial cell research here in Europe, so there would be a home to do that. So I think that this would be one of the problems is that the people who are working on these things, we are sort of say alone and scattered all over the place. We need to have some kind of focal point. And man, I mean, wouldn't that be good? I think this would be a great thing to have CERN to do this. So we're done. We talked a little bit about what life is. I told my version of how I believed it started. I've told you what we've done in the lab, what we're still missing. We talked a little bit, it was a little bit too fast. I was too ambitious. I wanted to tell you all these things and I didn't quite have time. But there is a big difference between origins and evolution. And evolution is not just evolution, there are different types of evolution. And I think very importantly, we should also think about what the implications are here once we create artificial living materials. And then I'll just say thank you very much for your attention, and maybe there are some questions.
Thank you very much, Steen, for this beautiful combination of physics, chemistry, biology, philosophy, many things. So time is open for questions. So for those of you who are online, please, you can either raise your hand or send your question in the chat. So I will read it for Steen. If I don't see any in the chat, so please, any question? Yeah, Dario?
So thank you for the talk. I should confess I was easily lost. A lot of materials that probably could be distributed in a set of lectures. But while listening to the initial part, we have all this diagram with the possible schemes for explaining what is the life cycle and other things. And immediately in my mind popped up the article by Anderson about the Morris complex. So clearly you have a colony of E. coli, and they are not interacting with the environment, and they stay what they are, and they try to optimize their life. But you are more than an E. coli, like much more. So all the cells of your body specialize to something different. How can we try to address this more challenging problem of trying to understand the diversification of the roles played by the cells? So could simply a simulation lead us to have something that is not just a set of cells that are growing, but cells that are specialized to digest, to interact with light, something that are doing something different. So something more that is closer to a life, not that just a cell.
So you mean so yeah, that I mean you think something that's more similar to you, or?
Yes, that is not just a colony of cells.
I don't know. I mean, it's of course wonderful to speculate about, but I mean, we don't even know how the Bau plan is of a cell. We know some part of it, and we don't know how to make a cell yet. What I'm proposing is something that's much simpler than a cell. And as again, I think one has to. I think it's a very good question, but one has to be realistic. I mean, it would be great also if we could have sort of interstellar flight and talk about that, but we can't do that. And so you somehow have to invest your scientific what's it called, energy in what is. I would like to do this before I die, right? But I'm not sure. I'm not sure how you'd address more complex life.
Well, again, when you had this example of the E. coli that were not interacting, they were finding some optimization. I was wondering if this colony, by the interaction with the environment, could have some subset of the colony that is specializing in some task, and the remaining part of the colony doing something different.
No, okay, I'm sorry. No, then I misunderstood. Yeah, no. Yeah, for you for Linsky and his team to teach these E. coli something else, he needs to introduce other species, or he needs to introduce more stuff, because these E. coli's they can only do what they can do. Okay. But if they're able to exchange genetic information from with other bacteria, or then you can get the things going. But it's there's some people, they don't like this story of or they're surprised that even a fully fleshed biological system like a bacteria can't evolve more than, I mean, it gets stuck unless it's in an ecosystem.
Okay.
Make a follow-up, because I understood that you could imagine, when you describe this optimization, it seems, okay, all these bacteria do the same and they optimize how to use these food.
As long as it's a single species, yes.
But now you could imagine that they specialize, they split up like a species split, and that they so that you have a colony A and type B, because they are using the available resources in a different way, and they are sort of so isn't there space in this colony for a kind of
I don't
situation in which
so do you talk about speciation? Yeah, I don't think so. If you are in one pot, I don't think so, because there will always be one that will be the one that dominates. But if you had a distributed system, so you had many different spaces, so then you could have things to develop. They could probably take different developmental pathways. But nevertheless, I think that if you studied any one of those, my bet would be that it would still be an optimization. I don't know, but that's my bet.
Thank you. First of all, thank you for your really interesting talk, especially for the last part, which are really fascinating, opening really fascinating questions. But my question is related with a conceptual doubt that I have concerning the idea of compositional information. And the thing is that on the one hand, as far as I have understood the meaning of the term by Lancet and these guys, which I think is that those that created the idea is that there is related with the composition that is maybe expressed in the sequence of catalysts. So the number of different catalysts expressed in the sequence, for example, of these components. Now, you have also mentioned in your experiment that you are playing with DNA. Mm-hmm. Now, DNA is an extremely inert component, which is precisely chosen by light because this low catalytic meaning or transfer, in the sense that it could explore the changes in the sequence in a decoupled way with the dynamic of the system. Whereas in the case of proteins or even RNA, changes in the sequence are immediately converted in catalyst differences, and therefore I think we manage two different views or two different forms of information. One is decoupled, and it's very important because it requires a kind of genetic code in order to express this information into dynamics, into catalytic functionalities, and the other one in more primitive forms of life or protocell infrabiological organization is direct.
Yeah.
So do you think that it means something, or
no? I think I have I have complete sympathy for what you're saying. I think that Well, first of all, see if I understand it correctly. You say that DNA has probably been selected because it's inert and it can be used for information storage, and these other, and it's not very well suited as a functional molecule. And obviously that's true. But at the beginning, before you had this bifurcation where you had RNA and proteins on the one hand and DNA as sort of the library or the recipe on the other hand, they must have, or they must, but I believe they started a similar place because it's simpler. And we're using DNA not because I think that's the best molecule to start with, but it's simply it's a pragmatism and it's money and ease of working with. And I'm a big fan of von Neumann that you know, whether we make it with one kind of molecule or another kind of molecule, for me it doesn't really matter. I would have preferred to not use modern biological molecules, but you know, sometimes you have to do what's possible. Okay. So I think that you're right, that we do have a...
That's why I have reminded the work by Grissemann and Salmari about the possibility to have a stoichiometrically freedom system in which you can, for example, invent a kind of regulatory system which requires this stoichiometric freedom from the sequence to the catalytic activity. So this has far-reaching consequences in terms of the evolution of...
Absolutely, and I would love to, if we had the money and the capabilities, I'd love to work with RNA instead of DNA. It'll be more potent. Or I think that PNA is also good because it has a hydrophobic, you can put a hydrophobic backbone on and you can synthesize peptide nucleic acid from prebiotic chemistry. So it's also a more interesting molecule. But again, it's too damn complicated. You can't, I mean, then the resources that you as an individual PI can gather, then you will have to just focus on that part. I'm interested in this systemic problem of actually having these the information and the metabolism and the container to play together, and therefore I have to cheat or to make it as easy as possible for us.
Okay. Yeah, so we have time for a very last question from the audience online. So it's Yvonne Santiago who is asking, so you have highlighted a metabolism first approach, and that information is a secondary step in protocells. What would be the role of mobility? Do you think that, sorry, mobility?
Yeah.
Motility.
Yeah, motility. Yeah.
So you think that self-motility or other transport phenomena could have already played a role in prebiotic conditions?
Absolutely, yeah, because you have motility. That is where you have in modern life. Then you convert chemical energy into mechanical energy, and you cannot have a cell division. You cannot have a container division without motility. So at the very core of self-replication, motility exists. And I think that, I mean, there's a lot of beautiful work by Hanzig and also Jitka from Prague. Hanzig is up in Padua, I think in northern Italy, where they've shown you can have droplets that can, by modulating the surface tension, you can have the droplets dance around and they can divide and they can follow gradients and whatnot. So it is a very fundamental property, and it's so fundamental that you cannot have division of an aggregate without motility or the fundamentals of motility. That's my opinion.
Okay, so well, thank you very much, Steen, for this beautiful talk. So let me just remind you that Steen will be here during next week too. So if you have any questions or any interest in meeting him, I'm sure that he will be glad to do so. And thank you very much again for being here.
And I'll also say that we will be back also the first 2 weeks of May. So for those of you who are going other places, there's another opportunity. And please help me or help us with the charge transfer if you can.
Venga, cuídate.
Apart from the replication strand, why does